All right, it should be recording now. Okay, so schedule. Today is the 27th, so we're going over Chapter 2. We will hopefully be able to get through all of Chapter 2 today. There is a lot in it. It's a pretty dense chapter, and then we'll cover Chapter 3 on Thursday. After that, your first exam opens. So your first exam is over Chapters 1, 2, and 3, and it's open from Friday morning, essentially at 12 a.m. So if you want to stay up really late on Thursday night and take it as soon as it opens, you could. But it's open for essentially 48 hours from Friday the 30th all the way till just before midnight on October 31st. So I know it's Halloween. It's definitely my favorite holiday. So I feel bad about putting a test on my favorite holiday, but it is a quick class. So it's really the best place for it. With that, there is a study guide already up on Canvas for you. So hopefully you're seeing me navigate to Canvas. So in our Unit 1 material on Canvas, hopefully you can see that right now. You'll see on my screen, Unit 1 exam is already there. On yours, there's no Unit 1 exam. Essentially, this is where you'll find it on Friday or Saturday, whichever day you take the test. So essentially, this little block will appear. You'll be able to click on it. It'll also say it's due in the material over here, the things coming up or due. And your study guide is right here. So I forgot to open the study guide. Let's open that, and I'll kind of explain that since this is your first exam. So the exams are multiple choice, entirely multiple choice. It's about 60 to 70 questions. All my exams are about that long. So let's open this, and I can give you a better sense of what the exam will look like. So I know we've only gone over Chapter 1 right now, so a lot of these questions you probably won't have any idea about, but hopefully you have read Chapter 2, so you'll know a few more. So these open-ended questions at the top, you can assume that there's going to be multiple questions, there's going to be a couple multiple choice questions on these top couple questions. So review neuron structure and function. We're going to talk about that today, the structure of neurons, what an axon is, what a dendrite is, the cell body, myelin sheath, those kinds of things. So what are the functions of all of those things? So there will be multiple questions about the neuron structure and its function. So you can kind of expect that from these. So they're kind of study questions because it's a study guide. And then these questions, the multiple choice ones down here, are pulled either directly from the exam or are very similar to the questions you'll see on the exam. So I won't send out like an answer key to the study guide. I definitely recommend you study in groups. You can meet with other students here in the Google Meet classroom. You can be like, hey, in the chat, you can ask people if they want to meet up, or if there's people that you've met with over the small group meetings that we've had last week. If you want to meet up with them here in this area, the Google Meet classroom, you could go over your answers to the study guides. If there's one or two that you're really confused about and you're not sure of, feel free to email me a day or two before the exam. So I guess maybe asking me on Thursday would probably be the best thing, maybe at the end of class or before class, because I can definitely clear you up on a couple of these, but I won't be going through the whole study guide. So again, this is the study guide. These open-ended questions, they're kind of just study guide questions. There'll be multiple questions on those topics, and then you might find this exact question, number 7, on the exam. I think students do a lot better when they know the format of the questions and kind of can expect what they're going to see. I actually might be able to, I'm going to turn off my screen sharing to make sure that I don't show you all the answers to the exam. But let's see really quick if I can actually show you what the exam will look like as you go to take it. And as I'm doing this, feel free to put up any hands or any questions in the chat box as well. Yeah, it looks like I can preview this. All right, so when you come to take this exam, I'll share that screen again. When you come to take this exam, this is essentially what you'll see. you'll see all of the questions basically in a really, really long page. With it being so long, do be careful. On some devices, if you click an answer and then scroll or use the arrow down, it can move the answer. So just be careful in your scrolling that your answers are not changing. So click an answer and then click out of the answer area, and then that won't happen. So I clicked out of it, and now I can arrow up and down. So just be careful none of your answers change unintentionally. You can flag answers or flag questions to return to them later. And when you do that, you'll be able to see them in this little taskbar over here. You can see a couple of these questions, but you'll see that you have a time limit for all of these. An hour and 10 minutes, I believe, is for this, and it is about 70 questions. And yes, it is all multiple choice. Yep, the whole exam is multiple choice. On some of these questions, it's like true or false, like this one. But other than that, there are generally 4 answer choices. So that is the format of the exam. So hopefully that gets you feeling a little more comfortable with the exam. And again, that opens on Friday and will close Saturday night. So any other questions about the exam? Yes, very good question. It is open book. It is open note. Yeah. So obviously, with a time limit, you're not going to have time to look up every single question in the exam, but it does give you the opportunity to, say, make notes beforehand. One thing I like to do before the exams is make like a note card of things that I don't really remember, or like in all my studying, I'm still having a hard time remembering or comprehending. So then during the exam, I can refer to that really quickly, just that note card. You can use a piece of paper or whatever. And that, when I do that, it's a really good study tool. So as I write down those things that I've been having a hard time remembering, I tend to remember them then on the exam. But if not, I can go through all the questions that I do know, and then maybe there's 5 that I didn't know that I flagged or something like that. Then I can go and look up those answers. So yes, it is open book and open notes, but with the time limit, don't expect to be able to look up every single answer as you go through. So yeah, very good question. Thank you for clarifying. I always feel bad for students who forget that on the first exam and they don't use their book and notes, and they might do a little worse than they would have otherwise. So thanks for asking as well. All right, if you come up with any other questions about the exam, let me know. For now, we're going to jump to the question papers. So here in the main Canvas page, you'll see in the syllabus and general resources, the question paper instructions, rubric, and template. So that is your biggest source of information for the question papers, as well as this discussion right now. It also has an example question paper right here that you can download. This is one that I wrote. You can use it as an example. You can see some citations in there. And then these are links to websites that might be helpful as you write a paper. If you've never written a paper before, an APA style paper, you might want to review these. It's not required, but all of these can help. APA cheat sheet basically tells you a little bit about how to cite common things. The paper template is literally just a document template for an APA paper. So it has basically a text box that says title, things like that. So you can fill in the title, you start filling in the text. This talks about how to paraphrase well. So as you read research, you will want to paraphrase that research. And so that will help you do that. How to read scientific papers. I'll kind of talk about that a little today, how to read the papers at your level. And then also quoting and paraphrasing, another basically cheat sheet. So again, these are all resources that could help you write your question paper, but these are the 2 primary documents you'll want to look at, the question paper rubric and the example question paper. So what is a question paper? Well, hopefully, as we are going through this material, as we're learning different things, you'll come upon, and I'm not going to read this entire document, I'm just pulling it up to reference a couple specific things. Hopefully, as we do about psychology, you find things that are interesting to you. So you might be really interested in the neurology that we talk about today, maybe specifically how the optic nerves are connected and how that's related to color blindness. Maybe you find it really interesting how neurons are related to our sense of hearing, or how neurons can be related to ADHD and how neurotransmitters work. So those are all kinds of questions that you might have leaving class today. Maybe after class last time, you're like, oh, I really liked those social psych experiments. That was some really interesting things. I want to learn more about social psych experiments, maybe even some of the unethical ones. What are other things the field is doing today to be more ethical? So if those are kinds of questions that have inspired you, this question paper gives you the opportunity to go and research them on your own, to take time outside of class to learn more about any topic. So the question paper can be inspired by class time, by the book reading, by anything that we discuss within the class sphere, but it should be related to the chapters, at least tangentially to the chapters that we've been reading. So in your schedule, you'll see that the first question paper is a midterm question paper, and it is due in middle of November. So with that, it can be on anything we cover from chapter 11, 8, 4, 3, 2, 1. So 8 is developmental psychology, 11 is social psychology, 4 is 4 is consciousness. Or maybe 4 is sensation and perception, and 3 is consciousness. One or the other. One is one and the other is the other. Today we're talking about neurology, and then chapter 1 was history of psychology and research methods. So as you go through these chapters, if you find something that's of interest to you, you should go and do your own research on it. So the final product, the final delivered product for a question paper is a 2-page research paper. I have an example up right now so you can kind of see that. So 2 full pages on the topic of interest that you've written based on your knowledge of this topic from new research that you've gathered. So you need to have at least, let me look at the rubric again, because I recently changed this, so I don't want to tell you the wrong thing. Let me look. Where's the rubric piece of this? 3 references. All right, so you need at least 3 references, and 2 of those need to be from a peer-reviewed journal article. So at least 2 full pages, double-spaced, using either Times New Roman or Liberation Serif font at size 12. That's about 600 words as a minimum. And with that, you also need a page of references. So these references that you read for your topic are related to your question. So if you're really interested in ADHD, you'll go and find research. And I'll show you how to do some finding of research in just a second. So you look for an article, a peer-reviewed journal article on ADHD and comorbidity with autism or something like that, if that's what you're interested in. And you find at least 2 peer-reviewed journals, and then you can have, and well, you need 3 references. All 3 could be peer-reviewed journals, but at least 2 have to be, and then the third can be like a reputable website. So American Psychology Association website might be good. Some medical website, depending on what you're looking at, might be good. Your cousin's blog, probably not the greatest source. There are researchers who do post their research findings in blogs as well as doing published papers. So that kind of case could be very useful, but you need to be able to determine if it's a really valid or valuable source in putting it as a source to your paper. So double-spaced. So do double-spaced, yeah. APA formatting, and APA basically stands for American Psychological Association. So essentially all psychologists in the US and a lot of other countries write in APA format. So, and the standard is double-spaced. Even though personally, I really like 1.5 spacing. I just think it looks prettier, not so many big spaces on a page. But for APA formatting, we're going to do double-spaced. My aesthetic preferences do not supersede the the learning of APA formatting. So this is an example. Well, let's show you that example paper again. So here you can see I've got my name up here, my topic, and my question that kind of inspired this topic. What are the possible negative effects of Ritalin? And so here I talk about, well, this is what Ritalin is, a stimulant. I'm not going to read this whole paper to you. So I'm talking about Ritalin is a stimulant. It's a fast-acting drug. These facts came from this source, Greenberg. And so, well, what is Greenberg? It has no date. That's what this ND means. So come down here to my reference list. It is a website in the APA, from the APA website. So it's a little paper I found through the APA website that I'm citing. So these facts that I'm referring to, oops, wrong thing. So these facts right here come from this source. So I'm basically paraphrasing chunks of that paper in my own words, and I'm saying, hey, I got this information from Greenberg. So here I have Ritalin influences neurotransmitters in the brain, including dopamine and norepinephrine. And that I got from Gottlieb from 2001. So who's Gottlieb in 2001? You can see in my references, it is another paper. And I'm not sure if that's actually a APA citation, so that might be a mistake. I should take some points off for myself. But you see here, I have another source from WebMD, another source here. So at least two of these sources have to be from peer-reviewed journal articles, and I'll show you in just a second how to find those. So essentially, through this paper, I'm talking about what is Ritalin, what are the possible side effects, and if you read through this paper on your own, you'll see that I discuss, well, why was I interested in this? Well, I was interested in this because as a adolescent or a child, I was diagnosed with ADHD, and my mother didn't want me to use Ritalin because she was afraid of the side effects. And so I'd never really looked into it, but I thought, hey, maybe I should look up what are those possible side effects. Now that it's years later, do we know more? And essentially what I found, and that's what this paper was on, is that there is the potential for stunted growth for someone using Ritalin. And it is a hard drug to take because you have to take it at very regular intervals. But other than that, the major problem that my mom was concerned about was that it would be addictive, like it would lead to other addictions to things like methamphetamines. And the research has shown that that's not the case. So as well as talking about the actual subject, I do include, at least in this paper, a little bit of, well, why I was interested in this. So you can do the same kind of thing. It doesn't have to be completely impersonal. You can write in first person, you can write in third person. Just make sure you're consistent throughout the whole paper. All right, so that is an example paper. It's the right length, appropriate number of references. This paper, the question paper instructions, has a lot more instructions. Like if you just read this entire paper, you would understand exactly what to do, but hopefully with me talking about it, you're understanding too. So it's worth 50 points, so it's about half of a test. And yeah, there's a checklist down here to be like, hey, make sure you did these things. Oh, and this is the rubric basically. So how do you find research? Well, the university makes it pretty easy for you. So if you go to our university library, so you can just click through to get to our library. This is its main page. You scroll down, you'll find this little icon that says research databases. So this is a really easy way to access a lot of research. So open up the research databases, and there are two that I recommend, although you can use more than these two. These are a good starting point. One is Google Scholar, and the other is PsycInfo. So let's see if they've got Google Scholar here. Sometimes they have it listed, sometimes they don't. So it looks like they don't right now, so I'll just open up Google Scholar in another. Google Scholar is useful because anybody can access it. You don't have to have access through a library, but if you do have access through a library, you will have more articles available to you than if you don't. So we'll go through that in a second. Let's look for PsycInfo. So I clicked on the P for the database listings, and near the bottom is PsycInfo. It says 1887 to current, because essentially what PsycInfo is a collection of essentially all psychology-related peer-reviewed journals, between 1887 and now. So you might not find something that was published like 3 months ago, but if it's published like 6 or 8 or 12 months ago, you should be able to find it through PsycInfo. So I thought I was logged in, but maybe not. You will have to log in. If you're on campus, you shouldn't have to log in because it'll know that you're on the same network as the library. So here is PsycInfo. Essentially what both PsycInfo and Google Scholar are a search engine for research. So let's pick a topic that we're interested in. Let's just use the same example we had before. Let's say ADHD and let's say Ritalin. So that's the topic I'm interested in. I click search. Let's say, nope, that's all I want to know. So I'll just click browse. Why do you do this sometimes? Sometimes it wants me to add. This is one reason I don't use PsycInfo very much anymore, because sometimes it comes up and it's like, hey, add extra things to your search so we find less, or that, so narrow down your questions. But I just want you to search those ones. There we go. Finally. Just had to put it back up in the search bar and search again. So I can go through these, and one thing that you'll want to do is make sure that you have academic journals, or it might be termed as peer-reviewed publications, as the source type. So we're going to say we want academic journals because it will pull up books or book reviews or other things like that. So you can see here, this is an academic journal, this is an academic journal. So you can also change your date that you're interested in. So let's say I want newer research. Depending on what you're looking into will really depend on the years that you can use for research. If you're talking about Freud, obviously some stuff that was published a while ago, decades ago, is still going to be relevant today. If we're talking about Ritalin and ADHD, probably in the last 10 years is going to be the most relevant research. So depending on the topic is going to depend on how far back you want to go. So I don't have like a year minimum, maximum. The newest relevant stuff that you can find is probably the best. So we see the efficacy of Ritalin in ADHD children under neurofeedback training. So that sounds like it might have a lot of good information about Ritalin as well as ADHD. So what I could do is click this full text. You can choose the HTML or the PDF. PDF makes it really easy to download. So I'm going to select that. And I do recommend you find and select ones that do have full text. You can select that as an option, so it will only give you things that the library has basically paid the fee to access. Essentially, part of your tuition fees for the library go towards providing these articles for you. So what I would do first when you see a title that you like, or you say, oh, that might be useful, open the article, and the first thing to look at and read entirely is the abstract. So read the entire abstract and see from that, does this sound like a relevant article? You might not understand everything in the abstract, and that's totally all right. But if it sounds like it's relevant enough, you might then jump into the introduction. So at your level, I don't expect you to go over the methods or the results, basically the stats section of the paper, but starting with the abstract, making sure it's relevant to what you want to look into, and then if it is, read through the entire introduction or even skim the introduction for information. So the introduction often has things like definitions of the concepts they're looking at, a history of research of the concepts. So it's going to be talking about in here about ADHD, things that are currently known about ADHD, where the field is now on studies of ADHD, as well as those same things for Ritalin, I would assume. So this is actually a really short paper, so you could probably read this one pretty quick, the entire thing. But again, once you get to this section that says methods, at your level, I would say just skip that. Don't worry about the methods. Don't worry about the participants, the design. Don't worry about the the results. So here they're talking about the results, their data analysis. You've probably never heard of a Mann-Whitney U-test for data analysis that they're talking about here. And since that doesn't make any sense to you because that's not at your level, you can just ignore it right now. What they'll do after their results in stats terms is they will have a discussion. And that is essentially where they will go back over those results in plainer English, depending on their writing style and what level they're trying to like write this at. You still might not understand some of the things in the discussion, but it should be just words. It shouldn't be stats here. So going to the abstract and then the intro and then the discussion is essentially all you need to do at your level. And that should give you all the information you need if this is a useful article. So again, abstract is a summary of the article. Intro is like the history of the topics, usually has definitions in there, talks about where the field is now and why they're doing the study. Discussion is plainer English of what they found. So, hey, we found this result and this result. This was related to this. Ritalin was useful because of XYZ, and they'll spell that out in a little bit simpler to understand English. So read through that, the discussion and conclusion, as well as the intro and the abstract. So a lot of this you would be skipping, and that would be totally fine. Hopefully you'll find a lot of what you need generally in the abstract and especially the introduction. The discussion, again, is their results. So that is the basic format of a research article. And let's say you were, let's say you wanted to paraphrase something in here. I'm trying to read as I'm talking, and it's not something I'm very skilled at. Okay, here this is saying, so alongside this usefulness, pharmacology therapeutics, so drugs for therapy, have several side effects. A number of children treated with stimulants either failed to show long-term improvement in ADHD symptoms or suffer from negative effects on sleep, appetite, and growth. So for my specific thing on Ritalin, that might be a good thing to cite, right? So it looks like, well, it could have problems on sleep, appetite, and growth. So in my own paper, I would write something like that. Previous researchers have found that certain drug therapies for ADHD can cause problems in sleep, growth, and appetite. So I'm paraphrasing this result that they're talking about in the page or in the paper. And so I'll actually pull up a document to kind of type that out and show you how I would write that really quickly, as long as it opens up quickly enough. Another thing I could do that I don't recommend you do a lot of the time, but you can do. Oh, it looks like I didn't save some documents. All right, well, that's not going to open up in time then. So another thing you could do is, if it's relevant, you could copy and paste a short section. Let's say they define ADHD or something. Let's see if they've got that. Attention to ADHD is a neurological developmental disorder characterized by symptoms of blah, 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 blah. So let's say I wanted to actually use this specific definition. I could copy this and put it straight into my paper as long as I cite it appropriately. There was a study paper I was actually writing the other day. All right, so let's say I wanted to cite this directly, that I wanted this entire quote for my paper. I think it describes it better than I could have, and I want to keep it exactly the same so I don't make any mistakes. I can do that, and I recommend you do that on things like maybe definitions, but you should be able to paraphrase most everything. Make sure in your paper it's in quotation marks, as well as has the in-text citation that you need and the page number. So year, page, so let's find out what those are. All right, so the authors here are Pactaman, Irani, and these other people. Really, when there's this many, If there's more than 2 authors, you only cite the first and say et al. I'll show you that in a second. But if you put all the authors, I'll mention it in a comment, but I wouldn't actually take points off for you. So we'll say this was written by this person et al. And then the year that this was published was 2018. So I'll put that in my paper. And it was on page, and this is the page of the journal. It might not necessarily be page 1. If you look here, it was actually page 2071 is where I got that from. I know that might be confusing. On the document that we downloaded, it was page 1, but what was that, 2071? So this is how to cite a full quote in your paper. So if you're quoting that definition, put it in like that. If I wanted to use the other piece that we read and paraphrase that, I would say something like, I'm going to not talk while I type because it's hard for me to do. Maybe it's the ADHD. What was the other? And growth problems. Sleep and growth problems. So that information, that fact, still came from this researcher. It's not a direct quote, but I still learned it from this person. So I would cite it in text like this. I would not have the page number. I would not have the quotation marks because I've paraphrased this. I've written this in my own words, but I still need to cite and show who this research came from. So there you go, an example of a direct quote and an example of a paraphrase. And you'll see those in my example documents as well. So my example question paper, I have a bunch of paraphrasing. Most of your paper should be paraphrasing the research you find. And I thought there was a couple quotes. Maybe I don't have the most recent one uploaded. I can fix that later. But yeah, that is what a question paper is. That's how to find research. I'm trying to think now if I'm missing anything vital that you need to know that I have forgot to say. Oh, let's search on Google Scholar really quick. So let's do the same thing, ADHD Ritalin. So one thing that you'll see is the results from PsycInfo. We found 279 that are full text academic journals. Let's see what Google found. So we found 24,000 results. So what Google is pulling from here is news articles, is books, is sometimes interviews with people, is a bunch of stuff, and any of that could be relevant. But when you're trying to find peer-reviewed journal articles, they're in there too, but sometimes they're a little harder to piece out from other things. So if you see this kind of thing, PDF at ResearchGate or PDF at AdVoices, you can be pretty sure generally those are there. Sometimes you'll find those for books where you can read the full text of a book, but generally this means that Google has access to the full text research article. So you can see here is the journal's name, Psychopharmacology. Here is Social Science and Medicine, and the author's names. So let's open both of these. I guess my point in showing that, was PsycInfo is really focused on journal articles. You can find them really easily there. And Google Scholar casts a broader net, so sometimes you have to click through more sources to make sure that it's the most relevant or the newest or something that's really useful to you. So you can find more through Google Scholar, and generally I use Google Scholar, but it can be more difficult. You sometimes have to sift through a bunch of junk. All right, so I would go to this paper, and I could read the abstract, and then if that sounded like a good paper, then I'd go to the intro, read that, the discussion, read that. Same thing for this article. So you can see here, this is, if you were looking at a printed journal, this is essentially what it would look like. You open to this article, and it would have these little icons on the top. It would have the title there, the author's name, then the abstract up here. So generally, a physical journal looks very similar to this. All right, so that is how to find research. Again, the library has a lot of other databases that are available to you. For our purposes in the psychology class, I would recommend PsycInfo as well as Google Scholar. Reading through the abstract, intro, and discussion should give you all you need at this level of the class. Any questions over finding research or writing these question papers? So again, if you go to websites, make sure they're a reputable website, something that is going to give you valid information. All right, well, I know that's a lot of time to spend on how to do a question paper and how to find and read research, but it is a skill that I hope you start to build, especially on something that you're interested in. So as we're going through different topics in the class, if you're interested in something that we talk about, but we don't dive into it as much as you would like, it might be a great topic for a question paper. If you're having trouble finding research, email me, let me know. Maybe I can send you a source or send you like a search link, essentially. So I can definitely help you through these, but the writing of it will be done on your own outside of class. And then you'll have two due over the course of the semester, one at the midpoint of the class and one at the final of the class. They're worth 50 points. Yeah, so 2 pages minimum of text, then a reference page, all in APA format. And then, oh, that is one thing I did forget, showing you how to find the citation that would go in your reference list. So here I showed you how to do the in-text citation, but I didn't show you how to get this stuff, the APA citation for your reference list. So in PsycInfo, it's really easy. It's actually easy in both ways. So if you're looking at the document, all you need to do is go over here to the right. Hopefully you can see that. It says cite if you hover over it, and it gives you all these different ways to reference it. Since we're writing in APA, make sure you're finding the one that says APA. Then you can literally just copy that reference and paste it into your paper. So where did our example paper go? So a APA citation has a hanging indent, which is basically just a fancy word to say the first line is like this and then all the other lines are like that. So this tells you all the authors of the document, tells you the title of the document, tells you the journal, tells you the volume and issue numbers, and then has a digital object identifier to make it easy for other people to find it. So this is essentially how you write a paper. You find good references. Well, first you have a topic that's of interest to you. You find some sources to teach you more about the topic. Then you write your paper and you include your citations in the text as well as in the reference list. So that is how to find it on PsycInfo is just over here on the right side. Click Cite. In Google Scholar, it's pretty easy as well. Let's say you chose this one. There's a little quotation mark down here. You just have to click that and it comes up with a bunch of different versions. So you can, again, copy this APA citation and paste it straight into your document. Technically, this one should be above this one because reference lists should be alphabetical. So it would actually look more like this, but with double spacing. So, all right. Hopefully that gets you started on these. And the biggest first question that you've got to decide is, what topic do you want to look into? So topics that students have looked into in the past are very varied. So if you're having trouble well, you've got a lot of time, maybe not a lot of time. You've got till middle of November to get these papers turned in. You can turn it in anytime in between now and then, but that's the deadline, is on November 13th. So if you get to maybe November 5th and you still haven't thought of a topic, email me, be like, hey, can you send me a list of topics students have done in the past? And I can send you a list of topics, and maybe one of those will spark your interest. All right, I don't see any hands or comments, so let's move on from this and start talking about neurology. I'm going to close a lot of these so my computer doesn't run as slow. Why are you not? Okay, that's why. You wanted me to save that paper. All right, so let's jump into chapter 2. All right, so with this, we are going to go over the nervous system, go over the structure of neurons, the structures of the brain, the endocrine system, which is part of the nervous system, brain plasticity, and genetics. Hopefully we get all the way to genetics because there's some really interesting things there, but let's get started. Let's watch this little video on mirror neurons to get started and hopefully get your brains working and excited for the topic after having so much discussion on how to write a paper for this class.
If you read some of the less reputable corners of the internet, you might have heard strange things about mirror neurons. According to some people, they are why we have empathy and ESP and telepathy. It probably goes without saying that most of that isn't true. They definitely don't give us superpowers, and there is conflicting research about whether they're the basis of empathy. Even if they're not, though, they are still a very cool part of your brain, and probably they help us learn from other people. The name mirror neuron comes from the fact that they fire both when you do something and when you watch someone doing something, even if you don't move a muscle. Basically, it's as if they mirror the neural activity of the person actually doing the thing. And they were first discovered in macaque monkeys in the early 90s. The story goes that one day, while a monkey had thin needle electrodes inserted into individual neurons in their brain, some of the graduate students, well, they went and took a lunch break. And while one of those graduate students was eating in front of the monkey, all of a sudden, there was neural activity in the premotor region of the frontal cortex in the monkey's brain. This region would normally fire if the monkey was moving, like reaching for food or eating. But those neurons were firing while the monkey was motionless and just watching someone else do those things. Further research started mapping out exactly where these neurons were, and they're remarkably specific. Like mirror neurons that fire when an experimenter grabs a piece of food wouldn't fire if the experimenter used tools to grab the food instead. But the big question was whether this happened in humans too, because these neurons were in regions of the brain where monkeys and humans differ quite a lot. Of course, your average human study participant is a little fussier about getting electrodes planted into individual neurons inside of their brains, so scientists used electroencephalograms, or EEGs, to look for mirror neurons instead, as they record neural activity through the scalp. Researchers had 20 subjects watch an experimenter demonstrate some motions with their thumb and index finger, and then perform the same motions themselves. And turns out some regions of their brains did act like the monkey's mirror neurons. Parts of the motor and premotor areas of the frontal lobe, for example, as well as where the parietal lobe meets the temporal lobe, an area involved in vision and focus focusing attention. But knowing that they're there and where they are doesn't tell us why they're there. Many neuroscientists now think that they help us learn from other people through observation. Take a 2009 study, for example, which looked at how these parts of the brain are active while people were being taught how to dance. The researchers used a game similar to Dance Dance Revolution and varied whether the participants were taught the steps using symbols on a screen or a human model in addition to the symbols. And they found that parts of the mirror neuron network really tune into human actions. For example, the superior temporal cortex was more active while watching a person demonstrate the dance than the symbolic breakdown of the steps. But perhaps more to the point, the participants' performance on the final dancing test was better when they had a human to watch, which suggests that these regions support observational learning. Human mirror neurons aren't exactly the same as the monkey ones, though. Ours seem to care about the actual movement, while theirs are more strongly tied to the movement's meaning. Like, say you grab a water bottle the way you normally would, and then you put it down and grab it with your hand like upside down instead, which makes drinking it a lot harder. It's basically the same muscle motion either way, but one serves a meaningful function. In a macaque, watching the meaningless action might not activate their mirror neurons. But in people, the mirror neuron regions of the frontal lobe activate regardless of whether the action makes sense. All that matters is the actual hand motion. It's possible that this helps explain why humans are so good at learning by imitation. Part of our brain is always paying attention to motions that seem meaningless, just in case we can learn from them. Now, you might be wondering where empathy comes into all this. Well, when you think about it, if a part of your brain activates the same way when you're observing an action as when you're performing it, then it isn't that far of a stretch to think that these mirror neurons could be involved in feeling part of what someone else is feeling simply by seeing them express an emotion. And although this would be very cool if it were true, the research to date is kind of all over the place. Like, one study did find that mirror neurons activate when people empathize with characters in scenes. In one scene, for example, the 18 subjects had to identify the emotions felt by both a son who was bringing home a failing report card and a dad who hasn't yet seen it, for example. And in addition to identifying the emotions of both characters in the story, they had to guess what both characters would be experiencing if they had more information. One of the mirror neuron regions, the inferior parietal lobe, was active while identifying the emotions in the story. And scores on a test of empathy were related to how active that region got when they had to make an inference about the dad who was still in the dark. Not all studies show this, though. In fact, many do not. But then again, most studies in this area have small sample sizes, so there's a chance that there are real relationships between these neurons and empathy that aren't being detected. And just to make things more complicated, these studies are all correlational. They can say that there's a relationship, but they can't directly infer causality, which means in the end, we don't know if these brain regions are necessary for empathy or if they just happen to light up when we're experiencing it for some unrelated reason. Like with so many things, especially in brain science.
All right, so hopefully that topic sparks your interest in neurons. So I think it's really interesting that humans have, as well as a bunch of other mammals, have these neurons that can activate when someone else is doing a similar thing that we could learn. So interesting, especially that humans seem to be able to do it maybe better than, say, the chimps, so that we can use our mirror neurons even when it looks like someone's doing something that doesn't really make sense, because maybe it does make sense and we just don't know it yet. So an interesting thing, but let's talk about what the actual neurons themselves are doing. So one thing to remember also is that these neurons, these mirror neurons, aren't in their own special part of the brain. It's just specific sets of neurons or clusters or networks of neurons that are activating in the same kind of area of the brain for doing these processes. So, for example, a lot of our motion for my right arm is controlled by about right here in my brain. So the mirror neurons over there might activate, even though I'm watching someone do this, they might activate, and it's in the same area as the real neurons that should be activating. So pretty cool, I think. So the nervous system goes beyond just the brain. It is the entire electrochemical communication system of the body. And so it includes the brain, the billions of cells and neurons there. It includes the sensory and motor nerves that go into our hands, our feet, all over our body. It includes our spinal cord. It also includes our endocrine system and hormones. So this is one thing people might not have realized before, is that our endocrine system and our hormones are part of our body's nervous system. So neurons themselves work quite quickly. So we can get a message of pain from our finger that goes up to our spinal cord and then up to our brain. And that happens really quickly, within milliseconds, versus hormones flooding our body system do have an effect on other aspects of the electrochemical communication across our body. They have an effect on the neurons and the processing there. They have an effect on other organ systems in our body, but they just work a little slower. Essentially, the hormones have to like seep into the blood and reach other parts of the body. So it takes a little bit longer to do that, versus the cells, the neuron cells, are pretty quick to send messages. So one big important thing to introduce and understand here also is the idea of plasticity. So plasticity basically means we all have the capacity for change, especially neurologically. So it happens because of a physical process of neurons themselves connecting and disconnecting and making reconnecting and making stronger connections actually in our brain. So does anybody remember the story about the taxi drivers in London from the textbook, and would want to share that with the class? Probably someone with a mic would probably be the best. All right, so the taxi drivers, they were studied and they were found to have certain areas of their brain that were much more dense with neurons than other parts of their brain. Does anybody remember what those parts corresponded to? So they essentially had more density, so more mass in their brain in specific areas than people who were not taxi drivers. So does anybody remember from the book where those corresponded to? Go for it, Shay.
Weren't they like better at reading maps than other people?
Yeah, essentially. Yeah, they had a mental map. It was in the area of their brain where you store mental maps. Yeah, exactly. So they were a lot better at mentally mapping the city or other places as well than just the general public is. So why is that? Is it just that people who can have that mapping ability, that they tend to become taxi drivers? Adam has a comment too. Go for it, Adam.
Oh, I was just gonna say, I actually work for DoorDash. So doing that for about a year now, like I've basically, like I know routes all over the place, like shortcuts and stuff like that, without needing to use like my phone or something. So, if that makes sense.
Mm-hmm. Yeah, perfect example.
Yeah.
I recently moved from Ogden to Sandy, and so I have none of those in my mental map. So I've driven with friends who have lived here for longer, and they drive all these little roads that my GPS has never taken me to. And I'm basically still stuck to following that GPS. So that's one thing that I would write for a question paper, is now that we're using GPSs so much and relying on them, are we doing a worse job at building those mental maps for the average person? I think for delivery drivers, I think the same thing is probably happening, because it's just more efficient to use your brain than to use your phone. So your brain has developed that skill. You probably have more density in those areas of your brain for mapping out your city. It's pretty neat. So Adam, did you have another comment still?
No.
Okay, no problem. And then I see another hand from Marissa. Go for it.
This just made me think of like when I was little, I didn't know like the streets and what they were called and like roads and stuff like that. my mom would always stress to me like how important it is to like know where you are and like what's around you and stuff like that. So like at this point in my life, now I've been driving for probably like 2 or 3 years, and it's like I feel like I know my way around everywhere. And I have friends who are just like, I'm like, okay, oh, you're gonna just get off on 72nd South, and they're like, I don't know where that is. And I'm like, how?
Yeah, really good example. So your brain is literally working in a different way, more efficiently than those friends that don't know these places and haven't developed those mental maps. And me as well, who also hasn't developed those mental maps. So I've got some good mental maps of, say, the area around Ogden, where I was living, but not here in the Salt Lake Valley. Everything is, all these directions are very strange. So really great examples. So what has happened for those of you is essentially that areas of your brain for mapping these things, these neurons, these networks of neurons connecting with each other, have gotten stronger. They've grown new axon branches or dendrites to make more connections. So Marissa and Adam, did you still have comments? I just hate to put anyone's hands down if they still have comments. Okay, perfect. So let's look at what a neuron looks like. We'll skip forward and then we'll come back. Oh, that's not what I meant to do. I was clicking too quickly. So this is a neuron. So what your brain is literally billions of these neurons that are connected up to each other. So each neuron is a cell. So here you have the cell body, and in there you have a nucleus, just like any other cell similar to a skin cell or a muscle cell. But the neuron cells are special in some ways. They have these little arms called dendrites. These are essentially your receiving antennas. Well, antennas maybe fair. It's not a great comparison, but let's just go with it. The receiving antennas, and then each neuron has only one axon. So they can have hundreds and hundreds and thousands of dendrites even, but only one axon. Axon is like the message highway to send a message to other neurons. At the end of that axon, you can have different axon branches. So there's more of these little spindly things. Like say my arm is the axon, so the message goes down my arm, and then at my fingers, this is all the axon branches. You can have more of those branches, but only one axon. So the neuron can receive an electrochemical message on a dendrite. It can be here, it can be here, it can even be on the cell body, or it could be here. So essentially, they're reaching out, these dendrites, to take a message from other neurons nearby. So for those of you who do know directions better, who have a better mental map, who are delivery drivers or like these taxi drivers, you've spent time and your neurons have shot out more of those dendrites to essentially create more mass on these neurons to receive more messages. The more messages that this can receive and the more axon branches it has, it means it's connected to more things. You could think of it like each of these neurons is its own. If you know anything about computers and networking at all, you don't need to know much. Yeah, you need to know just about as much as me, honestly. The neuron is one single computer, and it can get messages from many other computers as long as you have dendrites or Ethernet cables connected to those other computers. So you can then send a message to many other computers through cables. But specifically for the neuron, you have the cables of the dendrites that are bringing a message in. Then you have the one big axon sending a message out, but it can branch off. So you can think of one big message cord splitting off at the end and touching many other computers or neurons. So a couple different ways to explain that. I know I've kind of completely ignored this myelin sheath. Essentially, the myelin sheath is another cell of essentially fats that are put around the neuron that you can think of it as an insulation for the axon. It makes the whole process of sending a message speed up. And we'll look in a couple minutes in a little more detail about how the message is sent, but that is the basic structure of a neuron. You have messages coming in through the dendrites and then passing along down the axon to the axon branches and those terminals. So those of you who have those mental maps, you probably have way more dendrites and way more axon branches than people who don't have those good mental maps. And it's enough so that in these taxi drivers, you could literally see the difference in things like MRI scans, that area of their brain was more dense, that there was so much more mass because of those axon branches and dendrites. The human brain doesn't really grow new neurons after about age 2. After that, you essentially are only making new connections. There's a couple specific areas, and we'll talk about this in our chapter on development, that you can grow new neurons. But basically, after age 2, you don't grow any new neurons. All this plasticity is only happening from those axon branches and those dendrites connecting to new neurons and strengthening those connections. All right, so the brain is this whole system that integrates our senses, integrates messages from our organs, controls the processes of our body. Our memories as well are processed by these neurons themselves. We think, we make meaning and connection. Our brains are in that process. One of the main functions of helping us feel things like emotions. So yeah. So let's talk about a really basic neural pathway. And this is from an afferent nerve, from carrying information into the brainstem. And a good example of that is a sensory nerve, something like a pain cell or a heat cell in your skin. So let's say you get a message of pain. Maybe you got pinched or burnt or something like that on your skin. This afferent nerve is taking, you could think of these as its dendrites. With sensory nerves, it's a little different, like the neuron itself has a slightly different structure. These are they're activated differently than general neurons in your brain. Essentially, though, their dendrites get activated. It sends a message up past the cell body and then up its axon to the spinal cord, where it will go either to the brain, or that message could even immediately go down an efferent nerve, which is a motor nerve. So a motor neuron. So you see here, it's got these little dendrites. So each neuron is very, very similar, but slightly different function. Most of the ones in your brain are very similar to the one I showed you on that picture. Well, they look very different than that picture, but as you can see, this one has very short dendrites, this motor nerve, the cell body's here, and then a long axon going to the muscle cells. So you get a pain signal. The dendrites of this send it up to the spinal cord, to your brain, but it can also send it directly to a motor neuron. So it picks up that in these dendrites and sends it down the axon to the muscle. So essentially, this is the axon terminals, but rather than sending a message to other neurons, this is sending a message to the muscle to contract. So, I mean, there's not actually those muscles in your hand, but like your bicep or I don't actually know what the forearm muscles are called at all. Any of your muscles, though, they have efferent nerves going to them, telling them to contract through an electrochemical process. So if you get those chemicals, if that neuron at the axon terminal shoots out those chemicals into your muscle, the muscle contracts. That's what it does. Pretty cool process. Interestingly, plasticity is part of the reason that people who start lifting weights see generally a big increase in weight at first and then a plateau. So if you've ever decided maybe on as a New Year's resolution to start lifting weights or to get into shape, you might find, and you probably find actually, especially if you've been doing no working out beforehand, that you'll start out, let's say, bench pressing a certain weight. Let's say you start off bench pressing 45 pounds. Maybe really quickly within the next week, you can jump up to 50 and 55 pounds and even 60 pounds really quickly within the first couple weeks. But then after that, it kind of pauses. You start going up really, really slowly in your weights. So adding another pound every week, or maybe 2 pounds every week, or something like that. So why do you have that really quick jump? So it's actually because of neuron plasticity. So these axon branches can grow very quickly. They can actually grow and make another connection with the muscle within a matter of like 6 to 12 hours. So you're lifting, you go back 2 days later, you might even be able to already do a little more, not because your muscles have actually grown, but because there are more axon terminals telling more muscle cells to contract. So you've gotten stronger because you are telling more of your muscle cells to work. So rather than telling 10 of your 20 muscle cells to work, you're telling now 12 of your 20 muscle cells to work. Something like that, you know? And then once you get to the point where there's all the axon terminals are there, which can be pretty quick, you have to now wait for new muscle cells to be created. So new muscle cells, it takes a little while to get that process really starting and to actually see new muscle growth. A couple weeks actually, versus the hours essentially to get new messages to the existing muscle cells. So this is one reason we see weightlifters be able to go up in weight really quickly when they start lifting, and then they hit a plateau. And some people get really disappointed and disappointed when they see that. They're like, oh, I was going up in weight so quickly for so long, and now I'm at this plateau. I can't get past it. Well, no, now you're just kind of up to the point where your muscles now need to grow. So your neurons have done their thing, have done their plasticity. Your neurons are ready to engage all your muscle cells. Now you need to grow new muscle cells, or you should be in the process already of growing new muscle cells. So kind of an interesting effect of neural plasticity. And it's just because of those axon branches growing. So there's different sections of the brain. Obviously, we've got the spinal cord as well as the central nervous system. So our brain, we've got the hindbrain, which is this lower area here, the midbrain, which is kind of the middle stuff, like inside the middle of your brain, and then the forebrain, which is the kind of wrinkly outer portion of your brain. So each of these systems has specific jobs, and we'll go over them. And there is a ton more information that we're not covering. Here's a good video to get you started if you want to know a little more. We went over this. We talked about cell body. Well, I guess we didn't mention that it does direct the growth and maintenance of the cell. And that's a big important part of plasticity to remember, is that each of these neuron cells wants to live, right? It wants to do its function. It wants to be surviving. And the nucleus directs that, and the cell body is where the mitochondria are, things like that. So any cell wants to function and wants to function efficiently. That's a big part of our neural processes too. So it takes a lot of energy. Well, it takes energy for the cell to send a message, right? It grabs that message from here, it takes it down to this area, and if the message is strong enough, it'll send it down the axon. That takes some energy to move that message there. So if a cell is, say, not using this entire branch of a dendrite, or it's very, very rarely being used, there's no reason for it to keep feeding this branch of dendrites. So it can just delete that branch of dendrites right there, absorb it back into itself, and be more efficient, be using less energy to be as massive as it was, and be a little smaller. So if those taxi drivers, for example, if they stopped doing taxi driving over a little while, maybe even months for some of their cells, but on the MRI, seeing the density of their brains, it wouldn't probably be for a couple of years. You would see that dense area start to become less and less dense because these dendrites are being absorbed back into the cells. Axon branches as well, they can be reabsorbed. Oh, this is one thing I didn't mention earlier, is axons can be very long. In humans, our longest one is 3 feet long. Basically from our brainstem all the way down our spinal cord are our longest axons. Giraffes have some similar ones that are like 12 feet long. They go from their brainstem all the way down to their spinal cord. So they can be very long. The myelin sheath, since it is insulating the axon, helping that message speed up, it is susceptible to a lot of various diseases. One that I'm really familiar with is MS, multiple sclerosis. It essentially damages and hardens the myelin sheath and can lead to essentially signals being mistaken across neurons. So you could think of it as 2 wires that have their insulation kind of cut. It might cross messages, can do that kind of thing, or make the messaging in the brain a little bit slower. So it can make things like balance harder for people with multiple sclerosis, or walking and other things. So here is a really cool video that shows, well, I think it's really cool. It shows a human stem cell specializing into a neuron, and it takes about 14 hours to go through that process. So this would be in the fetal period of development, a stem cell becoming a neuron for the brain, and this can also happen up to about age 2. Stem cells become new neuron cells. But this will give you a sense of how quickly things like these axon branches and these dendrites can grow or be reabsorbed, and give you a better sense of, in reality, what they actually look like in your brain, because they don't look exactly like this picture here. I don't actually remember if there's sound here. It might just be music. All right, here we go. So there you can see the cells dividing into new cells. And now these stem cells are becoming neurons. So we now have a little neuron there. So obviously there's a lot that goes into that process that you don't necessarily need to know, like for the exam, but hopefully it makes you curious, or interested in what's happening to our bodies as we're growing, essentially. So there's some other videos on this that essentially show neuroplasticity. I'll show a little bit of this. It shows some examples of neurons that are already functioning and doing their thing, growing new branches for dendrites or axon branches. So hopefully you could see with their highlighted green arrows there, some of those axons, either axon branches or dendrites, kind of reaching out to find other connections. So that is essentially what's happening with neuroplasticity, that these different parts of the neuron can reach out and then make new connections and talk between the neurons. So I think it's a pretty neat process. So the next thing that we'll do is watch a video on what actually is happening at the molecular level chemically to send a message through neurons. So this is definitely more information than you need to know for the class. But especially if you already know a little bit about biology and chemistry, it might be really useful and help you make more connections. If it should help. I know for me, I'm a visual person, so seeing kind of what the process is doing at that microscopic level is useful for understanding how a neuron talks to another neuron. So we're going to watch this first one, and then the second one is a good one. If you're still a little confused or you want a little more information, that's another relevant one too. But we'll watch the first one together here.
How does the pain you experience when you burn your hand result so quickly in an action by your muscles? Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system. From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away. The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of...
All right, so a lot of chemical words there. Essentially what is happening is we're looking at the outside of an axon right here, and this is a cross section of the membrane. So here's the inside of the axon, and then down here is the other side of the axon, and it's just a cross section, right? So at the membrane of this axon, essentially what is happening is in a wave, we have the signal coming from a previous neuron activates one of these channels on a dendrite, essentially. And then the message moves down essentially like a wave. So if you've ever seen a sports game or a stadium where people do the wave, so they stand up, that looks ridiculous in the video. I'm trying to make my fingers do the wave and I'm not coordinated enough to do it. Anyway, essentially that's what's happening with the cell. So these channels or gates are opening up to let atoms that either have a positive or negative charge, that's what you would call these ions, is basically just an atom with a positive or negative, or a molecule, sorry, not just an atom, a molecule with a positive or negative charge into the cell. So as this gate opens over here, it makes this one more excited, and then it opens, and this one opens, and this one opens, and so on and so forth. So there's not actually any kind of ion traveling down, like it's not a single ion or like an electron or something. That's not traveling down the axon itself. It's that the axon itself has these membranes. So my fingers are the membranes, and so at the axon, it's opening the gates in sequence to change the polarization. And so it sends the message that way. So nothing's actually going down the axon. It's not like that ion is being sent through the axon. It's not like a little electron is being sent through the axon. So in that way, it's not like a wire. So a wire, like a copper wire, will send like a electron down the actual wire itself, versus this is just changing the chemical balance. My arm is now the axon. If you're wondering why my arm is up, my arm is the axon. So it's changing the chemical balance of ions outside the cell on these sides and inside the axon. And so it does that so quickly in a sequence. That is how that charge is sent down. So we'll watch this one more time. Hopefully that makes sense to you. Essentially, it's just a wave of these gates opening and closing in sequence to allow these ions in and out of the membrane.
Sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be reestablished. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, reestablishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly. At the axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse.
So this video, I think, is a good one that shows the kind of glow is like where the wave is. But again, it's saying these molecules, they don't travel very far. They don't have to travel down the axon. They just travel in and out of that membrane. So they're going a very, very short distance. So that's why neurons can fire so quickly.
As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly.
So you can imagine those gates.
At the axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse. The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels. The channels open, allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell. Because
All right, so I watched a little further than usual, but the end of that is really useful too. So essentially, at every point in these axons and dendrites, you have that series of gates to let those ions back and forth. The cell membrane of the cell itself also has that. So a message can touch, like this axon branch is touching this cell body. It could also be touching dendrites of other cells. And so that is how messages are sent through neurons. So it's not actually something going through the middle of that axon. It's the chemicals right here on the outside and inside very quickly going in and out of the cell itself. So hopefully that gives you a better idea of what is actually happening as our neurons are sending a message. I think it helps to understand how, at least at this level, how that message is sent, and then how they're structured and how they connect to each other, to understand a lot of the relevant processes that happen in our brain for things like forming memories or sensation and perception. So if you're still a little confused about what's happening there, this goes into more detail. Again, even more detail than you need for the exam, but another visual example, and they go more in depth on the chemistry in that video. So this is important because neurons in this process is how we think, remember, love, perceive, experience, create, and just generally live. This is how neurons are sending messages to our muscles to move. For me to flail my hands around in the little video box in the recording. So it's really useful to know, especially as we go to talk about, well, how do people think? How do people behave? A lot of it comes down to neuron structure and function. All right, so neurochemicals, there's a lot of them. Your book goes into more detail than we're going to go here. I'm going to put a slide up to say, hey, these exist. But honestly, for our purposes, knowing that they exist and knowing a little bit about them from your book is sufficient. So things like dopamine, you might recognize that as being relevant to happiness as well as addiction. Serotonin, we already mentioned that earlier today in relation to ADHD. So each of these neurochemicals do a different thing within our brain. So let's go to one that we've talked about. So norepinephrine, let's talk about dopamine. So dopamine is really relevant to a lot of things. So not just mood, it also is related to voluntary movement. Dopamine is relevant for sleep, attention, learning, recognizing rewards in the environment. So that's things like if you get a positive reinforcement, you might get more dopamine, for example. So it's related to extroversion. So people who have more dopamine tend to be a little more extroverted. So cocaine and amphetamines activate the dopamine receptors, tricking your brain to thinking that it has gone through an experience to give it more dopamine. So having too low of dopamine is linked to Parkinson's disease. So this is hopefully giving you, you might have known one thing dopamine was connected to, but it's connected to a lot of the functioning of our neurons. And as you see, there's this big list of neurochemicals. So, and this little diagram of what they affect. So each one of them affects more than one thing. So they have multi-purposes, essentially, is the main point I want to get at. And hopefully you can go through the textbook to get a little more detail on each one of them. With the time here in class, I think we'll skip over those. So before we go over how to study the brain, let's take a little bit of a break, and we will be back here at 4:10. So do a quick break. If anything has inspired you for a question, write it down. You can maybe write a question paper on it. But we'll talk about how to study the brain and other aspects of the nervous system when we come back from this little break. All right, and we are back from break. Hopefully everybody can hear me. If you can't hear me, please let me know. All right, so let's go on to talk about some different structures of the brain and how we study the brain in the first place. So the brain is obviously a really important part of our anatomy, and it's hard to access. So there's not really brain transplants like there can be for heart transplants. We don't really have computers that can replace the entire brain, right? Or artificial organs, like we can put someone who's getting a heart transplant on an artificial machine to survive during that transplant, right? So how do we figure out what different parts of the brain do? Well, one of the first ways we ever learned anything about the brain and its structure, its function, is from brain lesioning. So essentially, one way you can do this is by intentionally causing damage. So in a lot of lab animals, we have in the past created brain injuries. So specific ones, even broad ones, removing, say, like half of a rat's brain to see how it changes its behavior. So with that, for humans, usually that was from people who received injuries. So your book, I believe, I don't think I have it on a slide here. Nope, I don't. So you might in your book have the story of Phineas Gage, or that might be in a later chapter on personality. So essentially, Phineas Gage is a person who had a railroad spike go up through their head as they were working on a railroad. It damaged a lot of their prefrontal cortex, well, what we now call the prefrontal cortex, so this very front part of the brain. And their personality changed dramatically from before to after that happened. And so things like that, or if someone gets a really blunt force trauma like to the head but still survives, but all of a sudden can't see or can't understand language or something like that, that kind of gives us a clue of where in, or originally that gave us a lot of clues on what parts of the brain did what things. After a person dies, for example, you could do an autopsy and see, well, this part of the brain is clearly damaged. Maybe this is related to this problem this person had, the fact that they couldn't move their left leg or something like that. So we've also done this intentionally to animals in a more controlled environment to damage even specific clusters of neuron cells to see if, like, is this where their memories are being stored of how to run around a maze? So now we use things more like electrical recording. You saw in a video earlier today in EEG. Essentially, you're getting an electrical reading from those electrochemical interactions between neurons. So you can tell, like, the neurons here are now talking to the neurons here, and now that message is going to the very middle of the brain, and now it's going down to the spinal cord. So you can kind of track things that way. It is even so specific that you can now measure even specific single neurons. So brain imaging, I'm sure all of us have heard of MRIs, CT scans, PET scans. Essentially, a CT or CAT scan is a 3D X-ray of your brain, but that can be problematic to put that much radiation into your brain. So things like PET scans look at the glucose use by cells, so which cells are using more sugar, essentially. And then MRIs get a really clear picture of the interior of the brain using magnetic resonance and radio waves. So it's a little less damaging to the brain than shooting a bunch of X-rays at it, but it gets a similar picture. And then we also have fMRI, which looks at blood and oxygen use by the individual neurons, as well as the neurons, the supporting structures, because there's not only neurons in the brain. There, for one, the cells that create myelin sheaths and other neurons that kind of hold, other cells that hold neurons where they are. So looking at how blood and oxygen is used throughout the brain can tell you which part of the brain is active when you're thinking about your first kiss versus thinking about how to drive a car versus imagining yourself throwing a dart. Because different areas of your brain or some shared areas will be for all of those. So here are a couple brain structures to be aware of. And this would be a good example of something that I would put on my note card. So thalamus, hypothalamus, hippocampus. Honestly, even still to this day, I get hypothalamus and hippocampus mixed up just because the words look so similar to me. And I know they're near each other in the brain, right? They're near enough that I don't really distinguish them well. But the main point of this story about I can't remember words is it can be really useful to put that kind of thing on notes or a note card. So then you can refer to that during an exam. I would say, oh man, was that the hypothalamus or was that the hippocampus? Then I could look at my little note card and answer that on the exam. Again, exams are open book, open notes. Making notes beforehand on specific things that you know might be difficult for you is a really good idea. All right, so let's go evolutionarily oldest part of the brain forward. So this is the oldest part of the brain evolutionary wise, the hindbrain. So it's down here right near our spinal cord. It has the medulla, cerebellum, pons, and reticular formation. So being that this is the oldest evolutionary part of the brain, what kind of functions do you think it does? Or if you're looking at the book, what kind of functions are you reading that it does? So either put the answer in the chat or you can also raise your hand or just shout it out even. Turn on your mic and do that. That would be fine too. Go for it, Brianna.
It controls things like the heart rate, breathing, blood pressure, etc.
Yep, very good. So those basic survival functions that we need, like heartbeat, is controlled by this part of the brain. So it kind of makes sense, right? So this was one of the earliest things to evolve in the brain. It controls some of those most basic survival functions. So very good. The cerebellum, this area here, the very wrinkly part, helps control motor control, balance. So it's very active in, say, walking a balance beam, riding a bike or a motorcycle. So your brain stem is the thing that connects the spinal cord to the rest of the brain. So a lot of messages come through here as well. So as well as controlling those vital functions, controlling balance, the messages that are coming from these upper parts of your brain to tell your arm to move have to first go through this section to send that message. So a lot of basic survival, a lot of important functions here in the hindbrain. So between the hindbrain and the forebrain, so again, that forebrain is the big wrinkly outside part that if you just were looking at a brain whole, you would basically be seeing forebrain. Inside that is the midbrain. And so that is a lot of the connection between the hindbrain and the forebrain. It relays info from the eyes, the ears. So a lot of our sensory organs first send the messages here to the midbrain. Our eyes also send some of that message here to the cerebellum in the hindbrain. And then it has a lot of long axons going to all other sections of the brain because the information that it's kind of collecting is then sent to where it needs to get to, basically. So then the forebrain, again, is that large, big, wrinkly part that if you were holding a brain, you would be looking at. And this contains the limbic system, the thalamus, basal ganglia, cerebral cortex, prefrontal cortex, basically a lot of stuff. You can see a lot of those pointed out here as well. So here's the middle portion of the brain, and here's the hindbrain in this picture. So hindbrain, midbrain. So the limbic system, this does a lot of things for the memory and emotion. It has the amygdala in it, and the amygdala is important for survival, food, mating, social rituals, emotions. So as you can see, a lot of these areas of our brain do more than one thing. So the limbic system doesn't just do memory. It also does emotions too. You could think of it as this is where the Inside Out characters live in your brain. They're watching the memories and their emotions controlling it. We'll bring up Inside Out as an example a couple times through class because it's actually pretty relevant if you've seen it anyway. It's a pretty relevant way to connect with some of the topics. So the hippocampus. So this is one of those H1s that I get confused with. So directs memory, recall, and storage. But with that, the memories aren't stored here. So it's not like Inside Out where there's all these files of memories here in the hippocampus. The hippocampus kind of directs how to create memories. So you would actually find that your memory for, say, learning to drive a car probably includes some of the limbic system would be activated. When you remember the first time, you will activate some of the limbic system for that first time learning to drive a car. You were probably a little nervous or afraid, maybe a little excited. So those emotions will be activated in the limbic system. The motions that you might have been taking, maybe you were a lot stiffer, will be active in the motion parts of your brain. So essentially, the hippocampus directs different neural networks to kind of store that as a memory. It kind of tells which neural networks to activate to have you remember. And we'll go into a lot more detail in memory later on. But essentially, that's what a memory is a set of neurons, a neural network that activates, and it's about the same network and about the same neurons that actually activated when that event happened, which is really interesting. All right, thalamus is another relay station. It's getting senses, and I want to get to a couple of the other ones. I think we have enough time to go into a little more detail on these, but I just want to make sure. So the hypothalamus is really important, especially as we talk about motivation and emotion later on in a future chapter. So the hypothalamus monitors eating, drinking, sex, emotion, stress, reward. So this specific part of the body, of the brain specifically, is going to be really related to, say, dopamine as well, like we were looking at earlier, neurotransmitter information. But with that being said, this is called sometimes the pleasure center of the brain, but it's not the only pleasure center of the brain. There's other parts of the brain as well that control pleasure, that control the reward system, that have a lot of dopamine receptors and dopamine sent out as a neurotransmitter. So it's not the only one. It is involved in a lot of our emotions, in a lot of our day-to-day life. If you're doing things that you like to do, let's say you like to go out on motorcycle rides, the hypothalamus is probably going to be active during that activity. If you really like playing piano, the hypothalamus is probably going to be pretty active because you're getting that reward of enjoying what you're doing. If you really like cooking and really like sugar or something like that, you're going to be activating the hypothalamus as you're cooking as well as you're eating what you've cooked. So it's very often used. It's common to be active, essentially. It's not unusual to have a lot of activities that humans do activate this and other pleasure centers of the brain. So the cerebral cortex is the forebrain. So essentially, the big wrinkly part, the outside part, is the cerebral cortex, and it has the occipital lobe, temporal lobe, parietal lobe, somatosensory cortex, primary motor cortex, and frontal lobe. And in that frontal lobe, at the very front of that, is the prefrontal cortex. So you can probably recognize a few of these. So primary motor cortex, that's where a lot of the direction for where you move comes from. Somatosensory cortex, this is actually where a lot of sensations come into. So if you get a snowball thrown at your chest, you will feel that through this somatosensory cortex. If you get your finger burned on a stove, you'll feel that through this somatosensory cortex. The primary motor cortex would then be what you're activating to throw your own snowball at your opponent, or move your hand back from the stove. Like the video earlier, though, for something like a burn, that message doesn't have to come up to the motor cortex and sensory cortex. That message can just go to the spinal cord, which will send a message to the motor neurons to say, hey, just move the hand. So if you're getting burned or something, oftentimes you'll move even before you have any kind of conscious recollection of it or recognition of it. All right, so occipital lobe here in the back is very important for vision, which is nice when it's right next to this cerebellum, which is really important for balance. They've got a lot of connections between them. The temporal lobes here are for hearing, language, a lot of memory. So again, the specific memories themselves aren't necessarily stored here, but parts of that neural network are. So when I talk about neural networks, any neural network for, say, a memory, for example, or moving. Let's not go for memories right now. Let's say moving. So moving your arm intentionally to grab your cup of coffee. You probably have a goal, and a lot of that goal is coming from your frontal lobe. You have your primary motor cortex say, hey, start moving your arm. As you touch the cup or whatever, maybe it's too hot, maybe it's too cold. If it's too hot, you might send a message back and come here to this primary motor cortex or into your frontal lobe and say, hey, this is hot, I need to not grab it. Or if it's too cold, you might now make a new goal of, hey, I have to get up and go to warm this up. So let's say it's too cold. Mine's not actually too cold. I warmed it up at break time. But let's say that is the network. So I have this goal of I want some coffee. So I use my primary motor cortex. So axons here, or sorry, neurons here are sending messages down their axon, connecting with neurons in the primary motor cortex, which then send a message all the way down my arm to move. I touch the cup, and now I get a message back through my somatosensory cortex. And I say, oh, that's too cold. So there's neurons here connecting with neurons in the frontal lobe. I make a new goal to go and warm that up. So I might have to stand up. I need to be using the occipital lobe to be able to see and not run into everything. My cerebellum to be able to balance, again, and not run into everything. I have to remember things like how to use my microwave. So the whole network of neurons that I'm using is pretty big and connects through lots of areas of the brain. So a neural network, and that's when I say neural network, that's what I'm talking about. The whole set of neurons that are activating to do any specific kind of task, whether it's the task of smelling something or seeing something to the task of reaching a goal or driving a car or hitting your brakes before you get to the red light. So there's a lot of different neurons that have to activate in sequence in a network to do all these things. All right, oh, here's Phineas Gage. So Phineas Gage, this is a picture of him and the railroad spike that did go through his head. So without frontal lobes, humans are very often emotionally shallow, distractible, and sensitive, can't follow directions very well. Yeah, so that is what the frontal lobe does. That's also where the prefrontal cortex is, and that is involved in goal setting, for example. Reasoning and the executive control of what we decide we want to do. The somatosensory cortex, I've mentioned it a couple times. It's really interesting that specific parts of that somatosensory cortex correspond to specific parts of our body. So this funny looking little diagram here is essentially showing how much surface area or how many neurons are dedicated to each specific appendage there in the brain. So as you can see here, hands are really big and lips are really big. So if we look here, the lips take up a lot of area in our brain for sensations. So there's a lot of sensory receptors there. This is one reason babies put things in their mouth so much. because they have such good sensory receptors, and so much of their brain is dedicated to the sensory receptors of the mouth and lips, as well as hands. We have a lot of fine motor control, but we also have a lot of senses to our hands. So we can feel little touches, hard touches, heat, pain, cold, a lot of things. And so here, a lot of our brain is dedicated to sensing through our hands. So our trunk and our feet don't have as many, don't have as much compared to their size. Like the surface area of your hand versus the surface area of your entire trunk is very different, right? You've got a lot more surface area on your trunk than you do on your hand. But look how much difference there is in the dedicated area of your brain. I just think it's kind of interesting. So the motor cortex is essentially right next to that. So you've got the sensory cortex and then the motor cortex. So the part where you're sensing something that's happening in your hand is basically right next to the part that's telling your hand to move. And again, this is another funny diagram that gives you a sense of how much of the neuron space or how many neurons are dedicated to each and every process. So on average, there's a lot of space dedicated to moving fingers. This is why we have a lot of dexterity with our fingers versus there's very little space dedicated to our toes. So some people can learn, for example, to use their toes about as dexterously as fingers, for example, to play piano or draw. So to do like really impressive and detailed pictures. So what is happening there is essentially the plasticity of the neurons. You can, while this is kind of on average what brains are doing, you can have other neurons in this area dedicated to more dexterity in the toes or more dexterity in the fingers or more dexterity in the trunk. So I would assume if you compared the brains of people who did, say, gymnastics versus someone who doesn't really move their appendages, so let's say gymnastics and dancing, to someone who doesn't really move a lot of their appendages very often, there would probably be more cells, more neurons dedicated, and more axon branches and more dendrites dedicated to those areas of their body than someone who doesn't use those areas of their body as much. All right, so the corpus callosum is an important part of the brain, and this goes back to one of our first classes. We talked about right versus left brain. So here's the brain, and here's a cross section. We're looking at what would be a right hemisphere of a brain, and this part right here is called the corpus callosum. It's essentially a bunch of axons going back and forth. You could think of this as a whole axon highway connecting the right and left hemispheres of the brain. So it sends info to both sides. The right side sends it to the left side sends it to the right. And so it's where it's, you might not get this opportunity ever depending on what programs you go into and if you're doing medicine or psychology at another school. But I was able to essentially dissect human brains. And if you poke at like the corpus callosum, it's much more dense. Like it's harder. You can literally feel a difference poking it like with a measurement tool as opposed to this part. It was much more squishy. This was very dense as well, but it was really interesting to see that there is that much density of axons just all packed together sending messages back and forth. And this is one reason we say, well, nobody's really right brain or left brain because you're always using a lot of your brain. So you're not using 10%, you're using these neural networks of the area for balance, the area for vision, for memory, for goal setting, for motor control. So we're using a lot of our brain all the time. And those messages are passed back and forth through this corpus callosum. Interestingly, you can actually survive and function pretty well even with one whole hemisphere of your brain. I guess that would be 1/2 hemisphere of your brain gone. So as an example, some people who have really severe epilepsy, If those seizures are only happening in one hemisphere of their brain, sometimes the doctors, not as a first resort, more as a last resort, will cut this corpus callosum, sever that connection between the right and left hemisphere, and the seizures will stop. They won't be having that physical side effects of seizures. Some patients have even had a whole hemisphere removed and still survived just fine. A lot of the parts of their brain of the one hemisphere are then taken over by the other hemisphere because of plasticity. It takes a while to like get up close to previous performance on a lot of things, say language or reading, but people can do it. It's pretty impressive. All right, so the endocrine system is the other part of our nervous system that we often forget about. So the endocrine system are the glands that regulate a lot of our organs by releasing a lot of hormone chemicals into the bloodstream. So here are all of them listed, and we won't go into a ton of detail on each individual one, but we'll mention a couple. So hypothalamus is involved here. It secretes chemicals as well as takes the messages from chemicals in the bloodstream. Pituitary gland, pineal gland, thymus, thyroid, pancreas, adrenal glands. So for someone with chronic fatigue, for example, you might hear, have they got their thyroid tested? So because those hormones are related to sleep-wake cycle. So ovaries, placenta, testicles, adrenal glands, pancreas. I'm not sure I mentioned adrenal and pancreas. So all of these are also part of the nervous system. But these organs function by putting chemicals into the bloodstream. So glands are the organ or tissue in the body that create those chemicals. So we call them hormones, and those hormones control a lot of different body functions. So the hormones from the pancreas have a lot of effects on, say, the stomach and digestion. So the brain can send messages to the pancreas, or when the brain sees that there's a certain level of hormones, it might make you hungry. You might now feel hungry. And a lot of this connection between these glands throughout the body is happening in the hypothalamus. So it's got a big job there. So the endocrine system is a lot slower. So those neurons, they make those really millisecond connections with other neurons. The endocrine system, it has to pump those chemicals into the blood until it gets to a high enough level where the other organs start to activate or respond to them. So, but it is interconnected with the nervous system. So the pituitary gland helps control growth and can help regulate other glands in the body. The adrenal gland is on top of each kidney. It helps regulate mood, energy level, coping with stress. One of the main chemicals that it's involved with is epinephrine. So that helps you get more ready for action. So if you're thinking of like fight and flight, so these adrenal glands give you more adrenaline. So, and also secretes norepinephrine, which alerts other neurons and other glands. So it's a whole system communicating with each other, similar to how neurons communicate with each other in these electrochemical messages. The endocrine system can communicate with the brain as well as other organs, other glands, through chemical messages. All right, so the pancreas is under the stomach, and it's relevant to digestion, producing hormones like insulin is one of the primary ones that it's producing. It affects metabolism, how we're using glucose, how hungry we feel, which affects then our body weight. So when we're thinking about psychology, and that psychology is wondering and studying on how people behave and our mental processes, well, one of the ways we behave is by eating, right? That's a human behavior. A mental process is hunger, right? So the endocrine system and the pancreas specifically is part of that study of psychology because it's related to those behaviors. Hunger is one of those basic motivations. So we'll talk about that towards the very end of class in our chapter on motivation and emotion. So the pancreas is relevant there too. So ovaries are producing hormones for sexual development and reproduction, but not only that, they also develop secondary sexual characteristics. So the development of breasts and beards, that is also part of what the hormones produced by the ovaries and testes do. So it's not exclusively for sexual development and reproduction, but a lot of what they do is around sexual development and reproduction. So with that, brain plasticity applies here for the endocrine system, but when we're talking about it throughout class, usually we're talking about it in relation to neurons in the brain. And a key piece of this is while we've been talking about the motor cortex controlling movements, or the somatosensory cortex controlling the sensations that you feel at different parts of your body. If, for example, that somatosensory cortex is damaged, other parts of your brain can take over that responsibility. And so we're going to talk about 3 ways that this plasticity happens. We've kind of been describing it in briefly throughout the class, but we're going to go into more specific detail on this. And think back to the video that we saw on the rat neurons plasticity, that they were kind of reaching out new dendrites, because that's one of the ways plasticity happens. So collateral sprouting is one way brain plasticity happens. This is when the axons grow new branches. So my arm being the axon, it has this many branches. If it grows another set of branches, that is one way that the brain can do plasticity and use any healthy cells. So let's say you have 50 cells in your somatosensory cortex that help you feel what you feel in your hands. Let's say 20 of those cells are damaged by alcohol poisoning. Probably wouldn't actually damage that part of your brain, but that's okay. Let's just use that as an example. Or say a blow to your head, or maybe you fall off your skateboard while you're trying to do a trick. So you damage 20 of those 50 cells. So very often collateral sprouting will happen. The healthy neurons around those damaged ones are often activated by the same process, or are part of those same processes. So while you used to have 50 neurons, now you only have 30 that are doing the same thing. And so they might grow new axon branches to connect with the other neurons that those 20 damaged neurons were connected to before. So it's very often, but not always, part of that same neural network that's doing this collateral sprouting. Another one is complete substitution of function. So if a damaged region is damaged too entirely, its functions might be taken over entirely by a whole other section of the brain. And so an example of this would be if you had one of your hemispheres removed, parts of the right hemisphere would then take over, would substitute their function for some of the things you lost from that left hemisphere. And so this is very often also part of that same neural network. And that might make sense to you because as these neural networks activate, they're getting stronger. They essentially learn how to communicate with each other quickly. So if A is talking to B, is talking to C, is talking to D, if now it's A, B, C, A, B, and D needs to get activated, C2 might have been getting activated too, right? And so C2 might be on the other side of the brain, and so the brain kind of starts to take over that responsibility. Honestly, that explanation was probably just more confusing than helpful. Basically, parts of the network on the whole other side of your brain can totally substitute that function and take over the responsibility that you've lost from, say, losing those entire 50 neurons in that somatosensory cortex. So neurogenesis is when new neurons are generated. So this really only appears to happen in humans in the hippocampus. And so again, I totally just forgot. I'm mixing up the hippocampus with hypothalamus. So I'm going to literally scroll back to make sure I don't say the wrong thing about the hippocampus versus hypothalamus. Okay, so hippocampus is the memory one. Okay. Yep, so the area of our brain that directs memories being stored can grow new neurons. So this specifically in the subventricular zone. So new neurons can be created there, but it doesn't appear that they have a really big impact on human neurology and human brain plasticity. In rats, it seems to have a bigger impact than it does in humans, but we also can do much more detailed analysis of rat nervous systems than we can in humans. Also, possibly the olfactory bulb, which is relevant to smells and smelling things, seems like in humans it might be able to generate new neurons. So this is another way brain plasticity can happen, but it is not the most common at all in humans. It doesn't seem to be like the default. It seems to be a kind of very rare case that sometimes the hippocampus does make new neurons. Usually it's substitution of function and collateral sprouting that are happening to have neurons connecting with other neurons in the network, or other parts of the brain take over the responsibilities of those networks. All right, so let's talk about genetics and behavior relevant to the brain. So the frontal lobes are where things like personality, intelligence, voluntary muscle control, so that means like setting a goal to grab the cup of coffee. That is an example of voluntary muscle control versus the involuntary pulling back from the hot stove. So without these frontal lobes, humans are going to act very differently. So what is the relation between, say, frontal lobes and genetics? So remember, or in this class, we haven't really talked about this yet. So this might be the first time we're talking about chromosomes. So a chromosome is the thread-like structure in every human cell made of DNA. This includes neurons, this includes skin cells. So inside every nucleus of cells are chromosomes. So the DNA is a complex molecule. So with that, it's a long molecular chain made of a couple different molecules, and the way that is structured is how our body knows what to do with genetic information. So sections of that DNA are what we call genes. So proteins in our body read those genes and then do things like make your eyes a certain color, or make your pancreas produce more hormones versus less hormones, just on average. Proteins are building blocks of cells. They help run functions inside cells all over your body, and can even direct and run specific body processes. So I think the image is more useful for me anyway when I'm thinking of these. So here is a DNA. It's a really long molecule. The big long molecules of DNA are all wrapped up in each other to form a chromosome. But what we have is these DNA strands, and you see A, T, C, G are the molecular pairs. And so for these, any given piece of this DNA might be one gene, one message of the body to do something. So a protein, come over here, read that message, read, oh, this part says have more energy, or do more of this or less of this. And then it directs what the cells are doing. And that tells our body to give us specific eye colors, hair colors, all sorts of things. Sorry, I'm getting distracted by things outside my house. So what do genes do? Well, we have lots of things that the genes do. They send our bodies messages on how to create and regulate a variety of different functions. So we've got, sorry, a pizza just got delivered. Teresa, something is at the door. Yes, I know. I'm sorry. Towards the end of class, and there you go. Got ADHD showing. So since that time when I was diagnosed in childhood and adolescence, my symptoms are definitely better than they were before. But still there. Sorry about that. So proteins are a complex molecule. They're reading a message from the DNA, telling the body and telling cells what to do. So what does this have to do with our behavior? A lot. So let's talk about twin studies and adoption studies. So one way that we can try to see what behaviors and mental processes are related to genetics versus related to other things is through twin and adoption studies. So for a twin study, you take two people who are genetically the same. So they have the same genetic structure. If they're fraternal twins, it's mostly the same. And you compare the similarities in their biology, their brain processes, their mental processes, and their behavior, and see, well, how much of that seems to line up with them together, what's similar, what's different. Anything that's similar between those twins might be because of genetics. Anything that's dissimilar might be because of environment. So getting to that nature-nurture debate. So this is one way that helps us determine what behaviors and mental processes come from those genes versus outside things. Adoption studies are kind of similar. You take children that have lived, let's say you've got two siblings. Let's even say you've got two twins. So you've got two twins, they get adopted by separate families. That's a sad story, but probably also happy for them to be adopted though. So these two twins are raised in different environments. Then you could compare them later on and see, well, in this environment A versus B, what are the behaviors that are different? What are the mental processes that are different? Did this child develop ADHD versus this child did not? So does that appear to be genetic or does that appear to be environmental? So you can look at these things and compare twins, compare children that are adopted to in their family environment, like their original environment versus their adopted environment, to try and pick out and piece together what's coming from genetics and what is coming from the environment. So remember with all of these though, when we're talking about observing two twins and that their behavior is different in something but the same in something, that is a relationship there. It might suggest that some of those might be caused by genes, but it's not definitive experimental proof, right? When we're doing a study, we need to do an experiment to have that causal data. So a lot of these studies give us correlational data. Shows us that on average, I'm literally just going to make up a statistic here, but you could probably actually look it up. Maybe I actually should right now. So on average, let's say 44% of twins share the same kind of color blindness with their twin. So something like that, you might say, well, it looks like some of that might be related to genetics. But again, that's a correlation. That's a relationship between these variables, between the variables of genetics, between the variable of color blindness. It doesn't show for sure in an experimental, provable way and repeatable way that color blindness comes from those specific genes. So a lot of these, both the adoption studies as well as the twin studies, are correlational studies, not causal experiments. So let's talk about how they work or how they can work in humans. Passive genotype environment correlations, evocative and active. So passive genotype environment correlations is when a set of parents is probably going to have very similar genetics to their offspring, right? So let's say these parents are really good at reading. They have, let's pretend reading is a genetic trait, right? Let's just imagine it is. So reading quickly, let's say specifically reading quickly is a genetic trait. So they probably have a house with lots of books because they probably like reading more because they're really fast at it because of that genetic trait. So they have an environment that is already set up to support the genetic traits of the offspring. Let's say these people's genetics down here is better at music in some way. Obviously, playing music is related to more than just a couple genes, right? But just as an example for the class, let's say they're better at pitch. They can detect pitch better. Maybe that's genetic. I'm not actually sure. It's just as an example. And so if the parents are better at detecting pitch and better at music, they probably have more instruments around. Maybe they have a piano, maybe some percussion instruments. Maybe they're more likely to play with their children in that kind of way, right? So they have created an environment that passively supports the traits of their offspring. So that is a passive genotype environment correlation. So when a offspring or the individual doesn't change their environment themselves, when the environment is already set up in a way to support their genetic traits. Evocative genotype environment correlation is when the child themselves elicits a change in the environment. So let's pretend extroversion is entirely genetic, right? Let's say a child is extroverted, so they will probably be more likely to seek out other people and try to hang out with other people. And because of that, they will kind of elicit more friends, more social situations. Because they're being outgoing, they'll find themselves in more situations where they can be more outgoing. So because of their characteristics, the environment changes around them. So their characteristics makes the environment change around them. Active genotype environment correlations is a little bit different. This is when a child has those specific characteristics and then they seek out environments that support them. So let's go back to the pitch. Let's say having perfect pitch is genetic. So if you have perfect pitch and you're better at music, you might seek out music classes. You might seek out a music store or something like that, right? So you are going to pick environments yourself, intentionally go to them that are supportive of the traits you already have. And this can make it really difficult to study genes and environment. So this goes back to that correlation versus causation problem. So again, passive genotype environment correlations is when the environment is already supportive of the genes of the organism. Evocative is when the genes of the organism lead them to behave in ways that create a new environment. And active genotype environment correlations are when the organism is picking specific environments. So because of that, it can be hard to see, well, is this music aptitude? Is this because of genes? Or is this because of them being in an environment that was already supportive of music? So it can make it really difficult to distinguish what the cause of anything is. But we do have these correlations. And this goes even one step further for epigenetics. And I feel like I've skipped forward. So I thought epigenetics, okay, epigenetics is the last topic on chapter 2. For some reason, I was thinking it was at the beginning of chapter 3, because usually I have to do some of it in chapter 3. All right, so epigenetics takes this one step further. So let's go back to those slides on DNA and RNA, or DNA and proteins. So again, DNA is where our genetic code is, the organization of those molecules, that kind of ladder structure of the adenine, cytosine, thymine, and guanine, the ATCG. That is where we get our genes. The protein reads the order of that and then goes and does something in our body. So remember that when we're talking about epigenetics. So epigenetics is when our environment literally changes how those genes are then read. It doesn't change the genes themselves. The ATCG, the ladder structure, that stays exactly the same. That doesn't change, but it changes how it's read. So a baby might inherit genes, and then in prenatal development, maybe some toxins or healthy nutrition affect the way those genes are expressed. So affect the way that the DNA molecule itself is wound or unwound, so that when a protein reads it, it might read it a little differently if the DNA is wound more tightly versus less tightly. So it's about changing the molecular structure of the DNA strand, not actually changing the way the DNA is structured itself. It's changing the molecular structure because of environmental inputs. So your book gives a couple interesting examples. So the propensity for depression is on this gene, the 5-HTT-LPR gene, but it only activates when people have a stressful life. So in their genetics, the gene stays the same, the ATCG all stays the same. But when someone has a stressful life, that gene might then be read differently to be more likely to lead to a person having depression. So let's give a couple other examples. I actually really want to watch this video on it because it's short. Well, it's not super short, but it's really, the examples are good. So let's watch this video.
So say you have a clone or a secret identical twin, because you know that a twin or a clone is a genetically identical person to you, which is crazy, but it's true. Anyway, so you have this clone or your twin or what have you, and your parents decide that it's too much trouble to keep both of you. And so they give your clone to a kind of sketchy traveling circus. Now, I know your parents probably wouldn't actually do this, but just roll with me for a minute. So for the purposes of this example, let's just say that you grew up living a normal suburban American life, while your clone grew up with less stability and less access to nutritious food and less education, but more exercise and more access to sword swallowers and bearded ladies. And let's say you guys are 50 years old or whatever, you get together and have some lunch. So you're sitting there across the table from your clone. What do you think you see? Well, for starters, clone probably has a tattoo on its face. And if clone had stayed in the circus, clone probably, professionally rides a unicycle over a tightrope, so clone is probably in pretty good shape. But then again, clone probably smokes cigarettes and probably was malnourished as a child, so might be slightly shorter than you. By the same token, let's say that you've been eating Sonic tater tots 5 days a week since 1992, so you're looking a little bit tubby. Plus, you spent the last 20 years at a really stressful job that puts you at risk for all kinds of weird health complications. So at 50, you and clone would probably look pretty different from one another. But it turns out you don't just look different, you actually have become different. Now, if some scientists looked at your genetic code, they would say that your DNA was still exactly the same as that of the person. That is to say, if your genome is a paragraph, all of the letters would be in the exact same order. But on another level, there would be a huge difference. And if we extend this metaphor, we can say that the letters are in the same order, but the spaces and the punctuation are all in different places. Of course, completely potentially changing the message of that paragraph. The study of this genetic punctuation that I'm talking about is called epigenetics, which literally means above genetics. The epigenome doesn't change your DNA, but it decides how much or whether some genes are expressed in different cells in your body. Epigenetics looks at what happens to your genes over the course of your life and whether those changes could be passed down to your children or even your grandchildren. So here's the way epigenetics works. You have billions of cells in your body, and they each contain your DNA, the same exact blueprint of your genetic code. But just because they have the DNA doesn't mean that they know what to do with it. They need outside instruction from these little carbon and hydrogen compounds called methyl groups. The way these methyl groups control the genome is by binding to a gene and saying, do not express this gene. The methyl groups bind differently to your genome in a skin cell versus, say, a tongue cell or an eyeball cell. And that is one of the ways that a cell knows that, hey, I'm a skin cell, or hey, I'm an eyeball cell, or hey, I'm a muscle cell. cell. In addition to methyl groups, epigenetics is also controlled by histones, which are proteins that are basically the spools that DNA winds itself around. Histones can change how tightly or loosely the DNA is wound around them. If they're more loosely wound, the genes can express more, and if they're more tightly wound, then they express less. So whereas the methyl groups are more like a switch, the histones are more like a knob. Every cell in your body has a distinct methylation and histone pattern, and that is what gives every cell its marching orders. Think of your genome, the DNA, as the actual hardware of the computer, while the epigenome is more like the software, which tells the hardware what to do. The genome is what's going to be doing all the work, but the epigenome is going to be telling it what to do. So the hardware of your DNA is going to be the same throughout your entire life, but these epigenetic tags do change throughout your life, and they decide what genes get expressed or not. Now, epigenetic...
So let's go back to the slides really quick and go back to our page on DNA and proteins. So here, again, just as another way to say this, so the DNA here, the way it's structured, the A being the T right there, the C and the G, and so on and so forth, that order is what's then read by those proteins in, let's say, this specific chunk. So this specific chunk is blue eyes versus brown eyes. So these methyl groups, the way the DNA is wound, is then read by the protein in a specific way to then tell the body what to do, to tell the body more muscle cells or less muscle cells, all sorts of things. Also related to behaviors. So in addition to the proteins just reading the DNA as it is, the DNA has these tags on it, these methyl group tags, or it can be more tightly wound or loosely wound. Think of it this way. If the T and the G here should be read separately as a T and a G, but the DNA is wound super tightly, so then as a protein comes to read this, it might not read that T and a G there. It might just see that there's a T. Maybe the G is kind of overlapping, like the physical actual structure of that molecule is kind of overlapping itself. So the protein reads it differently. And why does this get wrapped up a different way? Well, maybe because of stress or because of proper nutrition or because of poor nutrition. So a lot of things that we do in our environment can actually end up changing the way these proteins read our DNA and lead to other effects. A couple other real life examples in this video, because some of those that I've given are kind of like silly, and in this video the face tattoo example is kind of silly too.
Information in a cell isn't permanent. It can change throughout your life, and it can be hereditary, and it can change over time, especially when your body is going through a lot of changes, like say during puberty. A bunch of methyl groups kick in and they're like, okay, so you guys over here, you're gonna have to start growing hair, and you guys over here, I really need you guys to get behind giving this guy some really horrible acne. Or like when you get pregnant, which hopefully I won't, but when you do, your epigenome has to be like, all right, guys, everything's about to get a whole lot bigger in here, and we're going to have to pass something the size of like a Merriam-Webster's dictionary through that little pipe over there, so let's get this thing done. But it's not just these dramatic times when the epigenome is changing. It changes subtly throughout our entire lives, and it changes based on a lot of environmental factors, like what we do, what we eat, what we smoke, and how stressed out we are on a daily basis. Scientists have found that things like a bad diet can actually lead to methyl groups binding to the wrong place and making mistakes. And with those bad instructions, cells become abnormal and become a disease. And then basically all hell breaks loose and you get cancer or something. Epigenetics is a very young science, though we've known about the epigenome since the 1970s. It's only in the last 20 years that we've even known what effect these epigenetic tags are having on our DNA. And even after they got all that business straight, scientists still thought that all of our epigenetic tags were stripped off of our genome before they were passed on to our children. So if you started smoking 10 packs a day when you were 10 years old, that would certainly be a horrible health decision for you, but you wouldn't necessarily be harming your unborn children in any measurable way. However, the thinking on that is changing pretty rapidly, because it's true.
So before we get into the idea of these epigenetic tags being passed on to future generations, which, I mean, spoiler alert, that's what he's going into there. Let's talk about a couple other examples. So we won't watch this whole video, but if you've ever played the game Pokemon, this is relevant to that. We're not going to watch the actually Pokemon part of this, but it kind of goes into Eevee as an example of evolving and changing because of epigenetic differences, in that it's never actually not an Eevee, it's just epigenetically different in a different form. So funny video, relevant to the class. We don't have time to watch the whole thing, and not everybody even understands what Pokemon is. Oh, that's a creepy picture. But there are a couple. There are a couple relevant examples here that I want to fast forward to. If it will give me this. Oh, there we go. So the honeybee is actually a really good example of a creature that can change based on epigenetics. So something that you and I can physically see because of epigenetic differences.
Parts of their DNA that can be unlocked by certain triggers in their environment. A great example of this are honeybees. Yeah, I know, I just dedicated an entire episode to bee talk as it relates to Peachette. Maybe I'm just going through bee movie meme withdrawals. I don't know. Bees are fascinating creatures, and in this case, they are the perfect Eevee example. So, like I covered back in that episode, female bee larvae can grow into either a queen bee or a worker bee. What makes the difference here is the food that they're given at birth. Bees given royal jelly become a queen, while everyone else becomes a worker. So in this case, royal jelly is basically the evolutionary stone for bees, determining what form the larval bee evolves into. It even changes the bee physically, where, in their adult stage, queen bees and worker bees look way different, but in their larval stages, they're practically identical. It's exactly like two different evolutions that wind up with two different appearances despite starting with the same looking Eevees. The queen and worker bees didn't start off as genetically different, but differences in their environment, the royal jelly, or lack thereof, led to wildly different gene expressions. And bees aren't the only place in nature where this happens. Another Eevee-like example happens in the agouti mouse, which has a gene which changes the fur of its coat. These two mice are genetically very similar at birth, but due to differences in nutrition, they have very different gene expressions, with one having a gene expression that causes it to become obese and have lighter fur. Same DNA sequence, different phenotypes, all due to environmental factors. In humans,
Well, let's actually watch this one human example too.
isn't just hair color either. Case in point, diabetes, specifically type 2 diabetes. We're often told two things about diabetes that seem in direct conflict with each other. First, that diabetes is a genetic disease. If your parents and grandparents had it, well, it's more likely to be passed down to you. But also second, that poor diet and lack of exercise can be a cause of diabetes. So then, which is it? Is it a genetic thing that you're born with, or is it a condition that you develop from eating one too many solo Vermonster challenges at Ben & Jerry's? That is a dangerous prospect, by the way. Do not try to do a solo Vermonster. Oh, the Vermonster, you are so good and so dangerous. The answer is it's both. You might inherit a genetic potential for diabetes that's part of the DNA sequence that you're born with, but that diabetes may only get unlocked if you have the bad diet. And if you maintain a good diet, well, you might never have to suffer the diabetes that was encoded into your genes, as those genes remain turned off. On the other hand, someone without a genetic predisposition for diabetes might never get it, no matter how many Big Macs they eat on the reg. And this kind of epigenetics perfectly lines up with what we know about Eevee.
So if you want to know more about Eevee as being an epigenetic example, go ahead and watch the rest of that if you know anything about Pokemon. But let's jump back to the SciShow episode on epigenetics, because I think those are really useful examples. So again, what is happening here is that these DNA strands are being read in a slightly different way depending on the environment. The royal jelly that those bees are eating, the stress that people are under, the diet that humans might make it so that this DNA strand is read in a way that leads to diabetes. So that is what epigenetics is. I think honestly those examples are clear enough. So we have these human environment correlations that a individual and organism can select an environment that is supportive of their traits, but our environments themselves can affect our traits as they are expressed by our proteins in our genes. So going back to this model that I've showed a couple times in class, you might get the sense that this will come up quite a few times, and it does, because an individual, a child, is affected by their environment. The environment can literally change the way their genes are expressed. So their relationship with their parent, if that's stressful or positive, maybe they have a secure attachment. That's going to change how this individual lives in later life. So a lot of things can affect our behavior and mental processes through life. Our environment can literally affect the way some of our genes are read through epigenetics. So like I've mentioned a couple times, you might start off with a propensity. Let's say the left side of this picture of these cascades is negative. So you might start off with that propensity towards depression, or that propensity towards having diabetes. But if you have a less stressful life, or if you have a healthy diet, maybe you actually end up over here without depression or without diabetes. So the things that we do in our environment can change our brains, our behavior. They can change the way we react to things. They can even literally change the way that our genes are being expressed. So it's kind of an interesting bidirectional interaction that we've only learned about in the last couple decades. So your book doesn't dive into this topic in as much detail as we do in this class. Again, feel free to finish either of those videos. But I think that is good for today's class. If anybody has any other questions, definitely let me know. And also, if you have any questions now about the exam, since we've now talked about Chapter 2, again, we're going to cover Chapter 3 on Thursday. Then the exam will open up on Friday. So let me know if you've got any questions. But I think that'll be it for today. Oh, I do want to say, I know we covered a whole lot of topics. We covered neuron anatomy, we covered brain anatomy, we covered genetics, epigenetics. And in some of the cases, I went into a lot of detail. In some of the cases, I kind of really briefly went over them. Definitely go back over your reading and your notes if you want to get more detail there. I've left resources in the slides, so go to those if you want more detail on those topics as well. But yeah, thanks everybody for being here at class today. I appreciate it.